Hello everyone. So today I'm going to be presenting on ATP production and regeneration. So throughout my biochemistry course, we had to make a poster for our lab, and we had to piece together different types of different pictures which all formed one big poster, and they all went together. And this basically is a video of the whole thing. So I'm going to take the next few minutes and I'm going to walk through the process of making ATP using glucose and CO2 as starting materials. So we will explore how nucleotides and ribose are synthesized and how they will come together to form ATP, which is essential for DNA replication. So, So, first of all, I want to begin by understanding the overall process of ATP. So, it is a primary energy currency of the cells. It consists of three main components: adenine, a ribose sugar, and three phosphate groups. The synthesis of ATP occurs through cellular respiration, specifically during the process of oxidative phosphorylation in the mitochondria. So basically, we're going to start ATP production. When we need to start the ATP production process, we need glucose and CO2, which we started with up here in our poster. So glucose is broken down through a series of reactions called glycolysis, which is about the middle of our poster. And so during the glycolysis occurs in the cytoplasm of the cell, and during glycolysis, glucose is converted into pyruvate and small amounts of ATP is produced. So next, the pyruvate enters the mitochondria, where it undergoes further processing in the steps called the citric acid cycle or the Krebs cycle, which I have all the way down here towards the bottom of my poster. See, all the way down, like right here. And so in the citric acid cycle, pyruvate is converted into acetyl CoA, which enters the cycle and produces high energy electron carriers called NADH and FADH2, which we have all throughout our poster. These carriers play a crucial role in the production of ATP. So now to focus on the production of the nucleotides and the ribose. So nucleotides are the building blocks of DNA and RNA, and ribose is a sugar molecule required for nucleotide synthesis. Both nucleotides and ribose can be synthesized through various pathways in the cell. So first, glucose is going to be converted into glucose 5-phosphate, which serves as a precursor for the synthesis of ribose. So, which we have up here, the start. And so, meanwhile, the synthesis of nucleotides begins with the formation of purine and pyrimide bases. These bases are synthesized through different pathways and requires various enzyme persecutors. Once the bases are formed, they are combined with ribose 5-phosphate to create nucleotides. So now that we have nucleotides and ribose, let's see how they come together in order to form ATP. So the process of ATP synthesis occurs in the mitochondria through a complex called ATP synthase. ATP synthase utilizes the energy stored in the high energy electron carriers, NADH and FADH2, to generate ATP. So during oxidative phosphorylation, the high energy electrons from the NADH and FADH2 are passing along a series of protein complexes in an electron transport chain, which is all the way down here on our poster. So as the electrons move through the complexes, they release energy, which are used to pump protons, or H+, across the inner mitochondria membrane, creating an electrochemical gradient. So this electrochemical gradient drives the protons back into the mitochondrial matrix through ATP synthase. As the protons pass through the ATP synthase, it causes the conformational change in the enzyme, allowing it to synthesize ATP from ADP and inorganic phosphate. So basically, to summarize, the process of ATP production starts with glucose and CO2. Glucose is then converted into pyruvate through glycolysis, which then enter the mitochondria and go through the Krebs cycle, or the citric acid cycle, and and then it produces high energy electron carriers. Glucose can also be converted into ribose 5-phosphate, which serves as a persecutor for the nucleotide synthesis. Nucleotides then combine with ribose to form the building blocks of DNA and RNA. So, the mitochondria are double membrane organelles found in most eukaryotic cells. They are often known as the powerhouses of the cell because of their primary function, and it is used to generate energy in the form of ATP through the process called cellular respiration. So finally, ATP is synthesized in the mitochondria through ATP synthesis, utilizing the energy stored in high energy electron carriers, the electron gradient generated by the electron transport chain.